to all students for joining for this lecture on the topic Periodontal Microbiology. Myself, Dr. Mohammed, Assistant Professor, Department of Periodontology at UBD College, Lucknow. The topic for today is Periodontal Microbiology, and we'll be discussing the history, historical aspect of the topic first, as who were the pioneers of the subject who discovered various microbes and finding gave various theories for the for the like, better study of the subject. Anthony von Leeuwenhoek called microbes as animal foods. J. Leon Williams they stated the significance of plaque. G. Lab coined the phrase gelatinous microbial plaque. W. D. Miller gave microbiology of human tooth. Weirhoff described the importance of bacterial plaque in the etiology of periodontal disease, and Lowe et al. Gave the landmark study on plaque. Roche gave modern theories of specificity, specific plaque theory, while Sopransky in 1979 gave modern version of specific plaque hypothesis, and so the others. Coming on to the oral cavity from micro's perspective, well, there are six major oral systems. Firstly, intraoral or supragingival on the hard surface. Second is subgingival region adjacent to the hard surface. Third is buccal epithelium and parietal epithelium and epithelium of the floor of mouth. Fourth is dorsum of tongue. Fifth is tonsils, and sixth is saliva. Fetus, as we all know, inside the uterus is sterile, and within 2 weeks it is nearly mature. Nearly mature microbiota is established in the gut of the newborn baby, and after more than 2 years, approximately 20 or 14 microbial cells are present, which is 10 times more than the bacterial cells in human cells. More than 500 different species are capable of colonizing the adult mouth, and at any individual typically harbors around 50 to 150 different species. Now, bacterial to host interaction comes in addition to a surface oral cavity on open growth system. Equilibrium or adhesion is equal to removal of forces. Shedding or high turnover rate is naturally occurring non-shedding surfaces. Junctional epithelium, AA and PG virus are bacteria, virulence factors, and endogenous bacteria. Now, mode of living, that is biofilm, it rarely find life so easily. Habitats, bacteria, biofilm interface, the microcolonies and water channels are mutually beneficial. Organic constituents of biofilm are polysaccharides, proteins, glycoproteins, lipids, and or material albumin that is present in GCL. Now coming on to dental plaque, WHO defined dental plaque as a specific but highly variable structure entity resulting from colonization of microorganisms on tooth surface, restoration of other parts of oral cavity, which consists of salivary components like mucin, scavenated epithelium cells, debris, and microorganisms, all embedded in gelatinous extracellular matrix. Gutmann also gave a definition. While dental plaque is a general term of complex microbial community that develops on the tooth surface embedded in a matrix of polymers or bacterial and salivary origin given by Philip D. Marsh. Then plaques are excellent example of biofilm. These are architecture of dental plaque. Biofilm has many features in common with other biofilms. It is heterogeneous structure, open fluid-filled channels running through plaque, extracellular matrix providing specialized environment and barrier function. As you can see in this diagram, that bacterial molecules and in between fluid channels over the pellicle, and pellicle is present over a tooth surface. Heterogeneous mosaic model, as you can see, is an extreme form of water channel model, which was given by Cable et al. In 1992. Their view of such biofilm is microcolonies from a start attached to substratum or subbase and are generally well separated from other neighbors. Now coming on to classification of dental plaque, it is supragingival and subgingival. Supragingival is foraminal and marginal, while subgingival is tooth associated, epithelial associated, or unattached. As you can see in this diagram, that 10-day-old supragingival plaque can be seen, which is the first sign of visible marginal gingivitis. Now, as you can see in the chart, that uh, gram positive is mostly dominates supragingival and gram negative mostly dominates the subgingival. Uh, and energy sources are generally fermented carbohydrates, whereas in subgingival many proteolytic forms of energy sources are available. Now, coming on to host cells in lingual plant, that is epithelial cells, macrophages, and leukocytes, intercellular matrix of dental plaque, organic constituents and inorganic constituents, and material from saliva, CCF, and bacteria. Coming on to composition of dental plaque, it is 80% water and 20% of solid. Other than bacteria, non-bacterial organisms include mycoplasma, protozoa, and viruses. 1 cc of weight about 1 milligram contains more than 10 to the power 8 or 10 to the power 11 bacteria. Intercellular matrix is estimated to account for the plaque masses consist of organic and inorganic material derived from saliva, gingival, cellular fluid, and bacterial products. Organic and inorganic solids approximately 20%, and water is about 70 to 80%, and inorganic it is mostly calcium and phosphate, and organic constituents of matrix include polysaccharide proteins, glycoproteins, and lipid matrix. Glycoproteins from saliva are an important component of the pellicle. Polysaccharide produced by bacteria, of which dextran is the predominant form. Albumin probably originating from pellicular fluid, while lipid material consists of debris from other membrane-destructive bacteria and host cells. Carbohydrates, mostly fructans and glucans. Uh, Fructans are synthesized in plaque from dietary sucrose, while glucose from sucrose and two types dextrans. The fats family as a skeleton of a matrix, and while plaque formation at ultrastructure level, the process of plaque formation is divided into three major phases: the formation of pellicle on the surface, adhesion, and attachment. And third is colonization and plaque maturation. As you can see in this diagram, the flow is from clean substratum to molecular to single organism to multiplication to substantial or absorption of the organism. Salivary pellicle can be detected on clean enamel surface within 1 minute or after the production in the mouth of volunteers. Or the pellicle is essentially in equilibrium between absorption and detachment. As you can see in this diagram, pellicle is composed of glycoproteins, proline, rich plasma, phosphoprotein, amylase. Significance of pellicle is protective in nature, lubrication, and digest for bacteria, and for calculus. As you can see in this diagram, that how the bacteria transport passively, and then after the receptor and attachment interaction, a reversible attachment is formed between the pellicle present on the enamel surface. Adhesion receptors uh, 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 identifies the secondary colonizer, and they form a coadhesion. Transport on the surface is due to Brownian motion, through sedimentation, through liquid flow, or through chemotactic movement. Initially, reversible adhesion is formed. Long and short range forces of van der Waals attracts at electrostatic repulsive forces 10 nanometer distance. Irreversible binding of bacterial cell to the pellicle and microbial cell surface are not uniformly coated with negative charge. Strong attachment between rough surface for beta production against shear forces, irreversible binding more easily and more frequently, substratum surface energy, removal of water film, short range forces. Large clumps of bacteria will not stuck well, removal by swallowing. A firm anchorage is established by specific interaction, direct contact, or bridging. Adhesins are subdivided into two major classes: spindrial adhesins and non-spindrial adhesins. colonization and plaque maturation. Primary colonizers provide new receptors for other bacteria, cell-to-cell addition with genetically distinct oral bacteria, clumps and co-aggregation, fundamental cycle, chemical forces, hydrophobic, electrostatic, and van der Waals forces. Post-bacterial interaction can be involved in bacteria, again, aggression and receptor. Due to this chart, this list shows various bacteria and their adhesions on receptors present in the oral cavity. Again, you can see how primary and tertiary colonizer attaches on enamel surface over a pellicle. This is again a plaque maturation diagram showing primary colonizer followed by tertiary, secondary and tertiary colonizer in the oral cavity and then surface. Co-aggregation forms a corn-cob structure, which is, for example, streptococci adhered to a filament of the Maturotrophy or Actinomyces species. Corn-cob is a very important share-side question as well. Well-characterized interaction, good co-aggregation, and these are the various examples of co-aggregation between two different species of bacteria. This is a very landmark article by Sopransky and Rastogi et al., in which they gave microbial complex for subgingival complexes and microbial complex and subgingival. As you can see, they classified various microorganisms depending on their severity into various color-coded circles. Red complex is mostly associated with bleeding on probing, whereas periodontal and non-periodontal infections are caused by green and orange complex bacteria. Dynamic of fungi plant, important changes in the plant growth rate can be detected within first 24 hours, during first 2 to 8 hours, adhering streptococcus saturate the salivary pellicular binding sites and first covering 3 to 30% of enamel surface instead of steady growth during the next 20 hours, a short period of rapid growth of about 4 to 6 hours. After 1 day, the term biofilm is fully preserved because the organization takes place within it. A bacterial density of a reaches approximately 2 to 6 million, and on enamel surface and mass increasing growth rate can be observed, while thickness of plant increases slowly with time, increasing in 20 to 30 micrometer after 3 days. During first 24 hours, plaque growth is invisible from a clinical growth point of view, whereas during night, the plaque growth rate is increased by 50%, possibly because of reduction in salivary. De novo supragingival plaque formation seen to surface in 4 hours is less than 3% covered with plaque. 3 days growth increases rapidly and slows down because of aerobic and gram positive. After 4 days, 30% would still be covered with plaque and shift towards aerobic and gram negative species like bacterium, filament, and spirochetes. It is technically impossible to record dynamic the subgingival plaque formation and established dentition for the simple reason that one cannot sterilize the periodontal pocket at present. Some early studies using culturing techniques examine the changes within the subgingival microbiota during the first week after mechanical debridement, reportedly on partial reduction, followed by the fast regrowth to the almost peak levels within 7 days. This is an experimental gingivitis model. In first few days, the plaque is mainly composed of gram positive, then later it is composed of gram negative, and which can be seen leading to gingivitis. This chart shows various factors influencing plaque formation, variation within dentition, variation within gingival inflammation, spontaneous cleaning, and variation within age. Topography of supragingival plaque. Plaque formation can also start from grooves, cracks, and pericarditis or fixed. Early colonization of enamel surface starts with surface irregularities mostly. As you can see, the dental area or other abrasions, etc., leads more to deposition of plaque. Again, you can see a treatment dye is surface micro roughness that causes threshold increase in pH addition. Characteristics of biofilm bacteria, the licence lancity, metabolism of large bacteria, nutrients present and saliva G-cell post-diet transition from liquid positive to gram negative, early colonizer uses CO2, and lower redox potential, gram positive uses sugar, gram negative uses amino acid and small peptides. Total plant population is more efficient, nutritional interdependency, critical to survival. This diagram shows more effective metabolism between all the primary and the secondary colonizers of the communication between biofilm bacteria or called as quorum sensing. Again, this quorum sensing is very important partner. And by the person, the bacteria that do not exist in isolation in a biofilm bacteria has the capacity to communicate with each other, that is quorum sensing. Evidence that soluble signals may regulate through growth of oral biofilm was proposed by L. of all in 97 types of signals molecules have been detected from lenticular peptides released by gram-positive organism during growth and a universal signaling molecule called auto-inducer. 2. Competence stimulating peptide increases the transformation frequency of biofilm grown. S mutant 6 to 10 to 600 fold and auto-inducer 2 AL2. Streptococcal peptides are known as competence stimulating peptides because the major response of these signals is the induction of competence and of microcycle physiologic stage during which cells are primed for DNA uptake and incorporation. Second quorum sensing system encoded by Lux's genes and uh, that is quorum sensing system respond to auto-inducer. In contrast, brain-specific competence peptide AI2 is produced by and detected by many different bacteria, including Pevingularis, Protozoa, intermedia. AI2 quorum sensing is closely linked to ability of the AA to grow in biofilm in an in vitro model as Spiralis, not a Oryx found biofilm manufacture. Quorum sensing gives biofilm distinctive properties, especially increase in for antibiotic resistance, influence community structure by encouraging the growth of beneficial species, discouraging the growth of competitors, physiological properties of bacteria, and the community may be altered. Antimicrobial resistance bacteria growing in a microbial community adhering to structure do not behave the same with the bacteria growing in suspended in a liquid environment. Resistance against the antimicrobial will increase by 1000 to 1500 times. An important mechanism of resistance appears to be the slower rate of growth of bacterial species in biofilm. Agents can fail to reach the deeper zones of biofilm by production of extracellular enzymes such as beta-lactamase, formaldehyde, and formaldehyde dehydrogenase. Antibiotic resistance may be spread through a biofilm by intercellular exchange of DNA. You can see how drug causes lysis of the microorganism in this diagram. This is diagram representing quorum sensing. This diagram represents a conjugation and transformation. Exchange of genetic information by conjugation and transformation, plasma transfer, and transposon transfer. Bacterial transmission of pathogens from one local to another. Vertical appearance of horizontal classification between individual. Bacterial translocation is from one niche to another. Full mouth disinfection has come into picture. Full mouth cleaning and reclaiming within 24 hours to reduce the number of salmonella pathogens. Tongue brushing front brushing with an antiseptic to suppress the bacteria and the image. Microbial specificity of periodontal diseases. Now, this is the chain view of various plaque hypotheses over the period of roughly 100 years. These are milestone and hypothesis in microbial physiology in last 100 years. Now, these are few of the hypothesis specific microorganism organism hypothesis. Second is non-specific plaque hypothesis and including mixed and anaerobic infection. Sufficient sufficient accumulation of any microorganism or below the general margin can reduce the destructive inflammation through the local production of enzymes. Specific plaque hypothesis given by Roche in 1979-92, red complex hypothesis given by Holt and Sopransky all at the distance of 10 years. All these hypotheses are again a very important short note. This is ecological plaque hypothesis given by Marsh in 2003. As you can see, the range between gingivitis and periodontitis from the general health. According to ecological plaque hypothesis, both total amount of dental plaque and the specific molecular composition of plaque can contribute to the transition from health to disease. Change in the host status such as inflammation, tissue degradation, or other high level follicular fluid may lead to shift in the microbial population of the plaque. This biosis is again a very important short note. This is gradually shift in the entire microbial community, which may result in chronic diseases such as periodontitis. Disease is associated with overgrowth of specific members of dental plaque biotin, which the local microenvironment changes, but which is not necessarily the same in each case. Keystone pathogen hypothesis is again one of the most important. Keystone pathogen hypothesis indicates that certain low abundance of microbial pathogens can orchestrate inflammatory disease by remodeling and normally benign biota in a dysbiotic one. On the basis, P. linearis was labeled as keystone pathogen. This means that it is an organism that is central to disease process and even is at a relatively low abundance. Now, another two is keystone pathogen concept and polymicrobial synergy concept, PhD, given by Clement R.J. This is again a very landmark study showing beyond the red complex and into more complexity and polymicrobial synergy in dysbiosis. This model shows PSD theory. Microorganisms associated with specific periodontal disease, mostly cocaine and rods are associated with gingivitis, whereas anaerobic organisms dominate the other. Periodontal is mostly anaerobic and very less associated rods and cocaine. This chart shows various microorganisms dominating various gingival diseases. Criteria for identification periodontal pathogen was given by Cohen, but Cohen as Cohen postulates association, isolation, reproduce, and recoverable. Sigmund Sopransky in 92 modified it for the oral cavity and gave to Sopransky's criteria. Virulence factor, a specific molecule that contributes to disease, factors that promote colonization, toxins, and enzymes that degrade host, mechanism that protect pathogenic bacteria from the host. Virulence determines those that promote attachment of pathogenic microorganisms. A is a small non-motile gram and stereotype B These are determinants of virulence and pathogenic potential of AV, and various factors are listed below. Again, in this, various factors and their activity are shown. Now, these are two various lab diagnostics or diagnosis for a strain. Each individual uh, is because it is green, or sorry, purple in color, production of porphyrin pigment in moderately fastidious growth requirement. Can be features gram-negative, obligate, anaerobe, non-motile, non-poring, asacrolytic, and cocobacilli depending on stage of growth. Culture grown in blood agar surface, usually white with red color. It has higher portion deep periodontal pockets, progressive deep periodontal lesions, infection, and failure implant. Variant structure, polysaccharide capsule, liposomal lipid is cysteine proteinases. These are two variant structure. Fingering major and minor finger binding saliva coated with hydroxyapatite particles in general, leaving certain fibroblasts, release of interleukin 1, 6, 8, and TNF alpha very protected for invasion. Lindy paints again very important for protection like protein, RGPA, and RGPA are very important, important vascular permeability management, inducing the dexterity through plasma like pre-calculin activation or subsequent bradykinin release. Pre-intermedia is another important, and these are various general and other unlisted. Ideal condition for growth is minimum and positive, brown black colonies or blood agar. Non-bacterial inhabitants of oral cavity are various viruses listed below. Papillomavirus that can be recovered from oral cavity is listed below. Retroviruses. These are two various new diseases caused by human herpes virus and periodontitis associated with syndromes. Ecological agents of different types of oral infection. This is fungal variations, contact oral bacteria and biofilms share microbial neighbors and build their productive mixed species to function with the signaling between bacteria and have important implications for the virulence of oral pathogens. It is mandatory that we know the taxonomy of the organism and various properties to establish a scientific basis for various diseases. These are the frequently asked questions on the topic. Thank you.